Good evening and good afternoon. Thank you for joining day 4 of our 2020 virtual user meeting, Cardiac Safety Testing from Ion Channels to Human Hearts. My name is Jason Villagomez, Marketing Manager at Nanion Technologies, and will be your moderator for today's presentations. By default, all participants are muted, and upon completion of the presentation, I will open the floor up to some questions. We encourage you to submit questions throughout the course of the presentations. It is now my pleasure to introduce Dr. Tamer M. Mohammed from the University of Louisville. Presenting Heart Slice Culture System reliably demonstrates clinical drug-related cardiotoxicity. I will now hand over to Amir for the start of the talk.
Yep, thank you so much for giving me the chance to share our research, which we actually, Nanion as well helped us a lot with the CardioExcite and this research. The problem that we still have so far is still in the 21st century, and we still have this problem where the current platform for testing cardiotoxicity, before clinical trials, you don't really go through any human tissue, any human heart tissue, I mean. So for the preclinical testing, we usually use either in vitro cells or recently human iPS-derived cardiomyocytes, or using rodents in vivo, or using in silico prediction. And it remains up to until phase 1 clinical trial when the drug touches the human heart. And this actually have led to make the cardiotoxicity is a major problem for drug withdrawals, and it become number 1 beside the liver toxicity for drug withdrawal or drug, or killing the drugs at phase 1 clinical trials. So, and these are just some examples of some currently available cancer drugs which have been marketed, and they are actually in the market, but they still have cardiotoxic effects. And we need to address these cardiotoxic effects somehow, or find some drugs that avoid these cardiotoxic effects. And as you can see here from this plot as well, that the cardiotoxicity does not only happen on cardiomyocytes. Sometimes it happens on the vasculature, and the peripheral vasculature, or the microvasculature in the heart, or it produces just arrhythmia, direct arrhythmia, by affecting the heart channels and so on. So this actually have led to the need for more reliable systems that could evaluate the cardiotoxicity in human heart. And as we all know that the human heart is not only cardiomyocytes. So of course the human iPS cardiomyocytes have made a breakthrough in this field, but it's still only one cell type. It's still relatively immature compared to the adult human heart. And the actual heart tissue includes a lot of fibroblasts, endothelial cells, and macrophages and so on. So to address this, we have done, we've just published recently last year, a system where we've been able to culture human or big heart slices of 300 micron under optimized medium, and optimize the electrical estimation conditions and the oxygenation as well, up to 6 days with full functionality as the fresh heart tissue. So this time window is enough to test acute and subacute cardiotoxicity eventually, or the effect of any drug on human heart tissue. So we've been able to record to do lots of readouts to assess the cardiac functionality. So we can do calcium transients. As you can see here, this is heart slice loaded with FLOO4, and they are stimulated, electrically stimulated. We could isolate adult cardiomyocytes from them and do electrophysiology recording. We could do as well, we can hang them in like an organ path and record the actual force and contractile function from them. And we recently adapted the CardioExcite to do this job instead of this tedious job for hanging them on an organ path. And I will show you some data from the CardioExcite recently. And then we can do metabolic flux as well, and we can do transcriptomic analysis, we can do electron microscopy, we could do immunofluorescence, And we collaborated with Tim McKenzie in Colorado, and we can actually isolate single myofibrils from these hard slices and determine the effect on the contraction mechanics and the relaxation mechanics of the isolated myofibrils after treatment with the drug. So how we do that, we just published the Jovi paper on this just to make this easy to spread, because I think our system is very easy to adapt to any lab, and we are happy to, to collaborate with anyone who wants to transfer the technology. We are spreading it over the US now, and I think many labs now are adapting this technology. So just to show you how we do this. So this is on pig. I can't really image human heart, but it's, so this is from pig. So we perfuse the big heart in situ first before we take it out. And then we perfuse it outside to just clear all the blood with cardioplegic solution. So, and then we dissect the left ventricle into cubes. So we dissect them into about 1 centimeter cubes to put them, just give us so we basically make them like 1 centimeter by 1 centimeter dishes, and then we can stick them to the holder, and we stick an agar to the holder, and then we put the cube on the holder, the agar, as you can see here. And then we start slicing them at 300 micron slices, as you can see here. So this is a vibrotome, so it's a vibrating microtome, which cut through the tissue at 300 micron thickness. And usually it takes about 10 minutes to do one slice. And then once the slice is finished, we take out the slice. And then we put it in this water bath with EDM in it, so it inhibits the contraction of it for a bit to relax the heart tissue. And with oxygenation as well, so we provide some oxygenation in this bath. And then we take the slice, trim it out, trim the edges, because the edges are not really good in culture, so we usually trim it to have equal edges. And then, so as you can see here, this is being trimmed. The slice is being trimmed. And then we stick some supports to the two ends, so we can keep the slices on, have traction on these small supports on the well, as you can see here. So we stick these supports, and then we put them, and then we put the slice in a 6-well dish. And then we take it. So now the slice on the support goes into the 6-well dish. And then we put the electrodes. So we use the CPS for electrical stimulation, for continuous electrical stimulation. And we use the C dish to stimulate the 6-well. So we put the cover for the C dish here. And then we put it in the incubator and connect the CPACE, and we introduce 1.2 hertz electrical stimulation of 10 volt for the 6-well plate. As you can see here, so now she's putting it into the incubator and connected to the CPACE stimulator. So, all of so there's no bioengineering materials in this system. It's very easy to adapt. So it's, so if anybody can basically adapt the system to his lab or his company or whatever. So why did we get into this actually? So, my experience is not really in hot slices. So I'm a cardiac molecular biologist. I've done lots of work on how to regenerate the heart using endogenous sources and using gene therapies. So I worked on direct cardiac reprogramming with Deepak Srivastava at the Gladstone, and I was the first scientist who started Tenaya Therapeutics, which is now a company. Now they are raising Series C. The first Series A was 50 million and Series B was 100 million, and now they are raising Series C to go into clinical trials to take this cardiac reprogramming towards the clinic. So now they are starting phase 1 clinical trial on some cocktails that we've developed. And then I recently as well published a paper that where we show that with Foster cycle factors, we can induce cardiomyocyte proliferation to regenerate the heart. So when we started at Tenaya Therapeutics, we wanted a system to test our gene therapies on a human heart. So we don't want our therapies to fail like others did when they touched the human heart in the phase 1 clinical trial. So we wanted actually to test it on human heart. So we wanted to prove that our concepts work. So induction of the cell cycle actually works on human heart. And this, what we showed here. So what we showed here is that we can induce with Foster cycle factors, we can induce the phosphorestin H3, which is a marker of cardiomyocyte division in human heart slices, as you can see here. But at this time, at 2017, so to decide to do that, we looked at the field, and basically at 2017, the field of the heart slice technology was using Transwell technology. I will go through it in a minute, but we wanted something to infect with virus. So nobody at that time have developed heart slice technology to infect with virus. So the first heart slice technology, or the first technology that been able to culture the heart slice for a certain amount of time, was developed by Brandenberger in Munich. So the group led by Dr. Dendrufer in Munich, they have developed this transwell culture where they expose the heart slice to air from the top and media from the bottom on a transwell. But however, this actually, even though they're still viable for 28 days, but the contractile function dropped massively by about 90% after 24 hours. So then there was some work from Igor Efimov here in Washington, where they developed, a similar system where they kept the heart slice on transwell as well for 24 hours as well. And then there was a lot of work from Tarsiano Group, where they have started to develop some other new submerged system to make the heart slice stay longer in culture. And it stays 2 3 days or so in culture, but not really perfect as well. There's some, fibrosis and so on. So So at that time, we wanted to produce a simple and reproducible medium throughput culture system to fully preserve the hot slice viability for at least 5 or 6 days to be able to test our gene therapy on them, because the virus expression takes about 24 hours to 48 hours to get expressed. So what we've done is we collaborated with Dr. Traciano Group to learn how to slice the heart and how to make 300 micron slices, and we learned it from him, and then we started to develop our own technology, but based on what we learned from him. So we compared the transwell culture with electrically stimulated culture. So we tried to introduce first the electrical stimulation. And with the medium that's been used all the time for research in hot slices, usually they use medium 199 with ITS. And this actually with the stimulation, with the electrical stimulation, didn't really preserve the viability of the hot slices. It dropped even earlier than the normal transwell. So we decided to screen for nutrients for the media to keep more the hot slices. And we ended up using, So we ended up finding out that the best media to use was the M199 ITS and 10% FPS and FGF and VGF. So this media provided the best viability for 6 days. So we kept the hot slices viable for 6 days with this media components and with the electrical stimulation. So we added both layers to the hot slices. And as you can see here from the calcium transients, we get the calcium transients at day 6. They are actually equivalent to what you see at day 0 from the fresh hot slices, and they respond to isoquinoline similarly at day 6 and day 0. But at day 10 and day 14, they really go, the calcium transients goes really irregular, and we start to see some dedifferentiation of the cardiomyocytes. In terms of contractility as well, day 0 and day 6 have had very similar stretch responses, twitch forces, as well as isoquinoline response, as well as the relaxation time as well was very similar. We've done RNA sequencing to look at the whole genome. It changes during the culture, and at day 6 we couldn't find any differences, any major differences, neither in day 2 or day 6. There's only 5 genes that's been differentially regulated at day 6 from day 0, but at day 10 we get about 500 genes that are differentially regulated, and mainly the downregulated genes were the cardiac muscle, and the upregulated genes were the fibrotic genes and the inflammatory genes. And as you can see here from these representative connexin 43 images, so the structure of the connexin is still very similar to day 0 at day 6. But however, if we do the unstimulated culture, the connexin expression goes way down as well. So we were not the only one who advanced this technology. There are the other groups as well who've been in this field for many years. They have done recent advancement as well on this technology. So Traciano Group in London have introduced a model where they stretch it. So they apply a preload to the hard slice, and this kept them for about 5 days with stretching them. However, there's some, if you compare to the calcium transients here to day 0, the viability is a bit lower when you stimulate them. And if you go to the calcium transients as well, the amplitude goes a bit up. And that's probably due to induction of hypertrophy due to the continuous preload that's been applied to this culture system. The Dendorfer Group in Munich as well, they have developed a system where they applied preload and afterload at the same time, where they hang the slice from one end and pull it from the other end. So make this free end to make the slice pull by itself back. And they have been able to keep the slices for 4 to 16 days, 4 to 16 weeks I mean, with reasonable twitch force kept in. However, if you look to the gene expression, there's some downregulation of the cardiac genes as well as early as 8 days. Ego, Efimov in London, in Washington as well, have developed a really like a heart on a chip system where they provide all the oxygenation and they can monitor EKG and contractile function of the heart slice. And actually they kept it for about 3 days. They kept the human heart slices with 3 days. However, if you look to the actual data on the map, on the heat map, there's a bit of loss in the edges as well, but it's pretty good, I think. It's pretty a good advancement in the field. So the major question after all of this advancement is, can we use heart slice technology to predict cardiotoxicity reliably over the available systems, over the available current systems or not? So to do that, we have chosen 3 different categories of cancer drugs that they can do. You know, they have some cardiotoxic effects on different aspects of the heart. So we choose doxorubicin as a positive control. So we all know doxorubicin exerts a cardiotoxicity on cardiomyocytes by inducing oxygen radicals. And we choose trastuzumab or Herceptin, and it's known to induce as well direct cardiotoxic effect on the cardiomyocytes. And we choose the sunitinib, which actually recently has been known to induce, hypoxia by targeting the microvasculature within the coronary vessels. So the sunitinib is being used as a test for the ability to show the effects on vasculature rather than the direct effect on the cardiomyocytes, because this is a reason that's been escaping the human iPS cardiomyocytes, because the human iPS cardiomyocytes are only cardiomyocytes and they don't have the vasculature layers. So to do that, we have choosing these 3 drugs. We did 3 dosages where the lowest dose is very close to the Cmax in the blood. And the higher doses just to see what was the further effect afterwards. And we assessed calcium transient, connexin 43, and we assessed the transcriptome as well. And as you can see from the connexin 43, in the control, we get a really good connexin 43 alignment. With the doxorubicin, we get, the connexin 43 being disrupted. Herceptin as well disrupt the connexin 43 and the troponin. Sunitinib actually didn't really disrupt that much the connexin 43 as well as the cardiomyocytes. So the cardiomyocytes are still a bit intact. There's really good connexin laid down here. Some of the cardiomyocytes get disrupted, but not that major. So the sunitinib actually doesn't have really major effect on the structural, at this dosage at least. There's no major effect at this low dosage, which is similar to the clinical levels, doesn't have really major effects on the cardiac structure. But when we look to the calcium transients, for example, as you can see here, this is the control media, and this doxorubicin. Doxorubicin, actually, there's really no calcium transients anymore because it's just killed everything. And then the Herceptin have induced some arrhythmic, calcium transients in the heart slice, as you can see here. And the sunitinib at this low dose have made some calcium overloading and reduced really the functionality of the calcium going around the heart slice. So then we used the CardioExcite as well, and we found that with the CardioExcite using the impedance technology, that all of these drugs at this dosage completely abolished the contractile function, as you can see here, and these representative traces, as well as the quantification. So does it transcriptionally match the changes, does the transcriptional changes match the non-clinical phenotype of these drugs or no? I think this was the major question of this quest. So for doxorubicin, for example, we know that it affects the cardiomyocyte directly. It's inducing the oxygen, the oxygen radical production. So we have done RNA sequencing on heart slices that's been treated with doxorubicin, the lowest dosage. And we found, a few hundred genes that been downregulated and upregulated. And if we look more in depth, what are the downregulated genes? You'll see it's all related to heart development and cell division, which means the direct damage of the cardiomyocyte itself and the nuclei as well. And the upregulated genes are mostly focused on oxidative reduction process and inflammation, which is what doxorubicin does actually, and we know that. So what about the Herceptin? The Herceptin is known as well to have a direct effect on cardiomyocytes and some on the vessels as well. So with the RNA sequencing, we found a few hundred genes are downregulated and some are upregulated. And if you look to the downregulated genes, we found that it's either the cardiac muscle contraction genes are downregulated, like what you see here from these three GOTERMs, as well as some angiogenesis gene as well being downregulated. And the upregulated genes are mostly translational, and this is known about the Herceptin that's activate the translational bit. So for sunitinib, the sunitinib is known to affect angiogenesis mostly. So for the downregulated genes for sunitinib, we found it's mostly focused on the blood vessels. And so it's blood vessel, erythrocyte differentiation, angiogenesis, all of these GOTERMs found in the top downregulated GOTERMs in this RNA sequencing. And as well as it's inducing inflammation as well and some immune response in the upregulation, in the upregulated genes. And in conclusion, I think that I've been able to show you that the heart slice culture system has got the potential to become a powerful predictive human in situ model for acute and subacute cardiotoxicity testing to close the gap between the preclinical and clinical testing results. And at the end, I would like to thank the group that they have already done the work, as well as our collaborators all over the country and the NIH and AHA for funding. Thank you so much.
Thank you, Professor Mohammed, for going through the presentation. I will now moderate some questions. So if there's any additional questions, feel free to add that in the chat box. Initial question that came in, can slices survive longer than 6 days?
So we are working on this. I don't have solid data yet, but keep an eye on our publications. Yeah. We are hoping to keep them for 2 weeks with some modifications of the media and adding, a correct preload and afterload to the slices. So we'll see.
And then a follow-up to this, did you examine the gene expression from the tissue or single cells?
Yeah, this is pretty hard to do single cell RNA sequencing on cardiomyocytes. Otherwise, we'll have to do it in the nuclei. So we haven't done that. But all the RNA sequencing that we've done was based on the whole tissue.
Okay. How did you fix the slices in the 96 well plate, and how long did you culture them?
So we usually, for the culture, we use 6 wells. We don't use 96. Do you mean the cardioid site?
Yes.
For the cardioid site, yeah. So for the cardioid site, we've tried different things to optimize it first. So we take punches of 96 wells. So we take a 6 millimeter punches from the slice and put it on the 96 well. We've tried different weights to put them on the slice to keep them flat, but actually without even any weight, we found it fine. And usually they don't last in these conditions for too long. So you have to do the recording within 10 minutes of making the punches and putting them in the 96 well.
And then could you induce the arrhythmias in the slices with rhythmogenic drugs?
Yes, this is something that we are doing at the moment. We are testing various compounds for 49 Therapeutics and for others as well that has got some arrhythmic potential. And we can decode. So we have optimized the way to decode the action potential as well as the calcium transients and see the effect of these drugs on these parameters.
Okay, perfect. Well, then if there's no additional questions, in closing, I will just say that I would make the on-demand copy of this presentation available latest tomorrow by morning time. And naturally, I will add any additional contact information for Professor Mohammed and the Nanion team regarding the technology discussed here, the cardiac site, and wish you a good rest of your evening and mid-afternoon.
Yeah, thank you.